Awesome. So we're a couple of minutes past now, so we'll get underway. And I just want to start by saying thank you all so much for sparing some time on a Friday to join us in this cellular conversations. The intention of this webinar, or this webinar series, I should say, is really to take a sort of dive beyond the biological story from people using our single cell or spatial transcriptomics products. So typically in a webinar, we get these fantastic biological insights where people have taken our tools and used them to do some pretty groundbreaking discoveries. However, the intention of this webinar series is a little bit different. It's meant to sort of go beyond that. We're really having a conversation about the experiment itself. How did you go about getting this great biological data in the first place? What are the tips and tricks and little bits of things that you learn along the way that you can help share amongst the community to make your experiments equally as successful? So very excited about today's webinar. I would say my role as a science and technology advisor means I'm having conversations constantly throughout the week with people who want to undertake single cell projects. And for those that are doing gene expression flex projects, a popular topic is the spike in of custom probes alongside our whole transcriptome probe set. So really, really excited for today's seminar, and I really hope that you get a lot out of this. So without any further ado, I want to introduce Dr. Jovana. She's a senior research associate in bioinformatics and computational biology in the Oshleck lab at the Peter Mac Cancer Centre. Her research is focused on epigenetics and transcriptomics data analysis with a strong influence in methylation array analysis as the co-creator of the widely used MIS-methyl bioconductor package. She was a lead computational investigator on a Chan Zuckerberg initiative, Pediatric Cell Atlas Network grant, contributing to the development of a globally accessible cell atlas of the healthy pediatric airway. Currently, she holds an NHMRC AL2 investigator grant and is focused on developing analytical approaches for multiple sample, multiple condition, single cell transcriptomics experiments. So with that, take it away.
Thanks, Jeff, and thanks very much for inviting me to share what we've been working on lately. And that's basically all about our experience with using 10x Flex to try and get a bit more interesting biology out of our samples using the custom probes that you can spike in. And it's specifically focused on cancer fusion, which is something that our lab has had an interest in for a while. So just to get us all on the same page, what are cancer fusions, if you're not that familiar with them? So basically, cancer gene fusions are caused by chromosomal rearrangements, such as translocation, and the sort of canonical fusion happens when you get breaks in the DNA, usually at introns that cause then rearrangement fusion of the DNA, and so then these fusion genes can then be expressed as RNA transcripts. And the sort of classic example of this is the BCR-ABL fusion, which is present in 95% of CMLs. And it's an important treatment target treated with imatinib. Whoops. Let me try and go back. Okay. Now, fusions are important because we know they drive the development of over 16% of cancers, particularly hematological cancers, and they're important treatment targets. We're pretty good at detecting fusions using bulk RNA-seq. So people have been working on this problem for a while, but they're really, really hard to detect in single cell, even though we're really interested in being able to do this. So how do we detect fusions in RNA-seq data? Well, basically, we're really looking for one type of signal, and that's if we've got our fusion gene here, we've got our two genes with a sort of breakpoint. We're trying to find either spanning reads, so reads where one side of the read maps to one gene or part of one gene, and the other side maps to part of the other gene, or spanning pairs when you're doing paired end, where you've got one of the pairs map here and the other maps there. And so, like I said, we've been doing this in bulk RNA-seq a lot, and so there's lots of tools that have been developed, and they've sort of gotten better and better over the years. And it's sort of only starting to come online now, a little bit for single cell, a couple of tools available, and some looking in the sort of the long read space as well. But basically, this is the signal we're looking for. You need to target the breakpoint because that's the thing that's unique to the fusion. So why is it hard to do this in single cell? Well, part of the reason is depending on what protocol you're using, you might just be missing information. So if this cat here is our fusion gene, where it's at two parts of different genes stuck together, and this is our RNA, then we're trying to target this break in the middle. And if you're doing 3' capture, So basically, you only get the tail. You can't target, you can't, it's very rare that you'd be able to capture this breakpoint. And the same with a 5' capture, you might just not be sequencing the bit that you need to be able to detect the fusion signal. Now, the other part of the problem with this is, okay, well, if we're not going to do 5' or 3', we need to do something full length, and then cost and throughput becomes a problem. And so if we look at the literature and what people have been doing, yes, there's methods to detect fusions at single cell, but they all rely on sort of full length protocols like SmartSeq or long reads. And the sort of numbers of cells that we're seeing in the literature are quite low because of this cost throughput trade-off. So hundreds of cells, maybe a few thousand in the whole experiment. So that's down to a few hundred per sample. And if we really want to dig deeply into biology, that's probably not quite enough. One of the reasons that, this is hard is because fusions aren't necessarily super highly expressed. And so, if we've got these 3 cells here, we've got our normal sort of genes expressed at various levels, and maybe we've got a few copies of these fusion transcripts. And this is the needle in the haystack that we're trying to look for. And, the tools that have been developed in single cell, basically they show very much reduced sensitivity for fusions with lower expression. So the more lowly expressed, the harder it is to detect. The other part of the problem there is if your fusion is only expressed in a few cells, so now we're looking at things that are subclonal, and just looking at this, so we've got our cells here, this schematic, and can you spot our fusion cat anywhere? If you've got eagle eyes, you can see there's one down here and one over here, and we might have put a third one in somewhere, but it's pretty hard to see. So if you're only expressed in a few cells, again, it's going to be much harder to detect at the single cell level than it is in bulk because we're not sequencing as deeply. So it's a pretty hard problem to get at in single cell. And so far, people have been trying to attack it with sort of doing really clever things in the lab or trying to come up with new ways to do sort of quite complicated analysis to try and sift out the real fusions from the noise if we pick up any. And we wondered, well, is there a simpler solution? And we sort of got this idea from the pediatric airway project that I was involved in. And so we were fortunate enough to receive this grant back in 2021 from CZI with my collaborators at the MCRI. And the idea behind this project was to make a cell atlas of the pediatric airway across 5 different tissues. And we were aiming to collect about 150 samples from more than 50 participants. And, we wanted to sample childhood starting from preschool to adolescence. So it's a lot of single cell samples, and these all had to be collected opportunistically, essentially, when someone came into the hospital. And so we really needed an effective way to kind of store and batch these samples as they came in so that we could minimize unwanted variation in the experiment, when we actually ran it. And back then, the sort of technology, the only technology that was really coming online to allow us to do this was 10x Flex, because it fixes the cells using paraformaldehyde when you collect them. So it sort of locks everything in, and you don't get issues like some cell types being destroyed by things like cryopreservation, which we'd had issues with in previous experiments. So we wanted to avoid that because some of the cell types of interest we really wanted to preserve. So 10x Flex was what we went for this particular sort of collection and batching problem that we needed to address with this experiment. But as we sort of got working with the tech and we started generating our data and learning more about it, we started to think more about what else 10x Flex was offering us because of its sort of unique properties. So yes, the fixing was sort of solved our problem that we had with the early air study, but we got really curious about the probe-based approach that this technology took as opposed to some of the others. So 10x Flex uses basically designed for a probe panel designed for about 18,500 protein coding genes, which bind to mRNAs, target mRNAs in 2 halves. So we've got two 25 base pair probes that bind the mRNA target, and then they're essentially ligated together. So if you get successful binding of both, they're ligated together, and then the probe is fished out by this sort of blue sequence that binds to the gem beads. And we're sequencing the probes here, which was a really interesting concept to us. And as we were sort of learning more and more about Flex, we discovered buried in the documentation that you could actually use custom probes with it. And that's when we started thinking fusions. Can we sort of, along with the standard gene panel, use custom design probes to now start picking up cancer fusions in single cell? So what we were really hoping to do was avoid all the fancy lab stuff and fancy analysis that people were trying to do, and do something quite basic. Basic in the lab, but also basic in terms of analysis, in terms of the sort of single cell analysis that we're used to. So what was this simpler solution that we came up with? Well, basically we designed this pilot experiment, which sort of involved playing each technology to its strengths, basically. So bulk, we know, is very good. We can sequence very deeply, and then there's lots of fusion detection tools to actually pick up fusion. So we wanted to leverage this to then allow us to design fusion-specific fusion probes that were specific to our samples that we could then spike in to our 10x Flex. And so we designed an experiment with 4 samples, one MCF7 cancer cell line, which has lots and lots of fusion and is well studied and is sort of the classic model for fusion biology. And then 3 patient samples provided by our collaborator, Teresa Sadras, who's now over at ONJ. And so 2 diagnosis and 1 relapse sample. And so we designed 24 different fusion probes for the MCF7 specifically for our specific MCF7 sample, and then 3 probes to probe the fusions that we knew existed in our patient samples. And all probes were used in all samples, and we barcoded each sample so that we could multiplex them in a 4-plex. And then basically all analysis, once it comes out of Cell Ranger, is as with normal single cell. One thing that we did have to think about was how do we design the probes to detect the fusion transcript. So if you recall, I said the Flex probes come in 2 halves. So they're 2 25 base pair halves that independently then bind to the target, and we're sort of relying on them then ligating together to detect the target that we're going for. So for a normal gene, what we have is our probe with our 2 halves here that sort of ligate in the middle at the 25th base pair. And when you have this ligation, then it's sort of counts as detected and it's sequenced and you get a count. So that's great for normal genes, but if you remember at the very start, I said the key signal that we're looking for in detecting fusions is this breakpoint. And so we thought, well, this is the thing that's unique to the fusion, but we don't really want to put a probe half either side of the breakpoint, because we were concerned that that would affect sort of specificity, and that then each of the probe halves would also be competing for binding with the wild type gene. And so we decided early on that we were probably going to have to offset the breakpoint into either one or the other side of the 2 halves. So you can see sort of an example here where in this case our fusion breakpoint is located here, and we're sort of offset at least. Well, the documentations from 10x recommends that you introduce at least 5 mismatches to help with specificity. And so that was sort of our lower boundary on designing our probes, that it had to be at least 5 base pairs into the left or the right side of the probe. We didn't want it too close to sort of either end. And then we needed to think about some of the other factors that were going to affect probe binding in terms of optimizing the design. So we looked at the documentation to think about what else we needed to consider, and the docs recommended one of the first things was, the ideal to aim for is 3 probes per target, and most of the genes in the panel, in the standard panel, have 3 per target. However, with our case of the fusion genes, that's not really possible because we're trying to target the breakpoint. So we knew that we were really only going to have, we were only going to be able to have 1 probe. And so we wondered how we were going to go with that. And because we were really interested in the flex technology, we had a master's student actually deeply digging into the probes and the performance of the flex probes in various in our flex early air data, but also in some publicly available data from 10x. And so Calandra looked at the various aspects of the probe, the standard probe panel. And what she found was that yes, indeed, most genes had 3 probes. However, there were lots of genes that had to have, either more than 3 probes or less than 3 probes. And there were about almost 1,000 genes that did only have 1 probe. And looking at their expression levels across several data sets, this is just one example here, they didn't seem to be doing significantly worse. And so that sort of gave us a bit more confidence that even though we could design only 1 probe, we were probably going to be able to detect what we wanted. We had to think about the GC content. That's obviously very important. If anyone's ever designed primers, we know that that's, something you really have to think about. And the third thing that was sort of noted in the documentation that we really had to think about a lot was this 25th nucleotide must be a T. So this is what the documentation said. And we found that to be quite restrictive for us because you can imagine, where we need to target this breakpoint and we need to be slightly offset from it so that we can get specificity in either the left half or the right half of the probe. And we found that when we included this constraint, a lot of probes sort of failed. We didn't test them, but in a computational sense, they weren't going to be successful. And so so we asked Calandra if she could look a little bit into this or in the probe set and see, was this T really a deal breaker, and could we sort of loosen this criteria a bit? And what she found in looking at the data was, again, yes, T was sort of ideal. Most of the probes did have a T there, but there were a few with other bases. And that, their expression was sort of reasonable. And so based on her looking at the data, we decided that we were going to relax that a bit and maybe, allow it to be either an AAC or a T, but maybe avoid Gs. And turned out that, what she found in the data was exactly what 10x confirmed for us in a later conversation we had. So that was really great. And we also looked at homopolymers, repeats, low complexity sequences, SNPs, and things like that. And our RA, Victoria, who was basically building the probe design software, she sort of built in all of these heuristics into the design process. And so we've now got a little, software package that can kind of design fusion probes for us for Flex. So ordering the probes, once we had our probes, from our software, then we had to go and order them and spike them into our 10x panel. So like I said, we had our 4 samples, and as I mentioned earlier, for this pilot experiment, we basically decided to include all of the custom probes across all of the samples so that we could get some idea about specificity, because we knew that the MCF7 fusions shouldn't be coming up in the patient samples and vice versa. So it was kind of a nice little internal control. And so we designed 4 identical opals, opals 1 to 4 for probes against the MCF7 fusion and the leukemia fusions, and these basically only differed now by the addition of these unique barcodes for the four samples so that we could multiplex. Now, these were just ordered from IDT as opals, as shown here. And we did initially try for this experiment the recommended concentration, which you'll see did work. But we've since optimized this in a subsequent experiment, and we'll show that it works much better at detecting fusion transcripts in primary samples when we used about 4 times the concentration of the custom probes based on advice from Andrew at 10x. So the nice thing about the custom probes is that they're basically just added to the experiment during the probe hybridization step. So from what Teresa tells me, that part of the protocol was quite simple, and there wasn't anything particularly difficult about adding the custom probes into the normal panel. Okay, so now let's get on to the results. From this experiment, the data looked really good. We were so very excited. And it pretty much looked like ordinary single cell, where each of our fusions basically came up as an additional sort of gene in the output. And as you can see here, we got, several thousand genes per sample, which is much more than the examples that I showed previously from some of the protocols that people had tried previously to detect, where they were trying to natively detect fusions in single cells. So this was super exciting, and all the quality looked good. And the most exciting thing was that we could actually see fusion expression. So I'm just showing some examples here of the different fusions that we designed probes for in the MCF7s. So you can see them expressed at different levels in the MCF7 cells. And what we could also do, which you basically couldn't do this before, was that basically now we could see which fusions were being co-expressed in the same cells. So we could see that on average, MCF7s were expressing, or at least we were detecting the expression of at least 5 to 6 fusions per cell on average, but some had many, many more than that. And when we looked at some examples down here, so this is just a heat map showing the cells here on the X and a couple of the fusion genes here on the Y. The BCAS3-4 fusion is a classic one in MCF7 cells where the expression of the fusion is higher than each of the wild type genes, and you can sort of see that here. And looking at our patient samples, again, the data looked great. This is our UMAP here of our patient samples, and then mapped to the bone marrow reference with azimuth. We can see that, we've got quite a few different cell types represented, but mostly the sort of pro-B type cells, which we expect in leukemia. And then if we zoom in and just tag our fusions, we can sort of see them here in the different patient samples. And we can see that, again, they're mostly the cell types that we expect, but we do see them come up in some other cell types, which is very, very exciting for Teresa in particular. So some interesting biology coming up there. As you can see down here, the numbers that we were detecting, however, were sort of quite low, only tens to hundreds of cells expressed where we were detecting the fusion expression per cell. But what was encouraging was that even at these sort of low number of fusion cells detected, we were sort of seeing the expected pattern of expression. So on this expression plot here, you can see our 4 samples. We obviously don't see any of the patient fusions expressed in the MCF7 as we expected. We see lots of expression of the CRLF2 fusion in this CRLF2 control sample that we included. And then we can see these fusions changing in expression as expected based on the bulk at the single cell level. And what was most exciting here was that in this relapse sample here for this patient, we were seeing the expression of this fusion down to the level of 5 cells, I think. Yep, it was 5, and this was not detected in the relapse sample at the bulk at all. So we're showing some really good sensitivity there. And we managed to do it again in a second experiment. So this is now in more patient samples, all BALL with ETV6 Frunx1 fusions. So we ran 2 fourplex runs. And as I said before, we this time included 4 times the recommended probe concentration. So you can see here that for, again, each of our samples, we're getting lots and lots of cells, so more than 10,000 cells per sample, apart from this MRD one down here, which had a very low input number. So this wasn't due to the experiment or anything like that. And, you know, the quality also looked really good. So we were very pleased with this. What was most encouraging, though, was when we looked at the sort of the numbers of cells that we were now detecting the fusion in. If we zoom in on our pre and pro B cells, which collectively we had, you know, tens of thousands of these cells, you know, 60,000 of one and 20-odd thousand of the other type, you know, we're seeing expression of the fusion in maybe around 40% of these cells. So quite a significant boost in the numbers of cells that we're seeing the expression of this fusion. And we do have some ideas about what kind of analyses we could do to see if, you know, there are other ones that we're maybe missing, and if we can also somehow group them with the fusion expression cells that we've now tagged reliably with the probes. So just finishing up with some more examples of the expression of the fusions in these BALL ETV6-RUNX1 samples. You know, you can sort of see them marked here on each of the samples in this diagnosis and relapse pair from this one patient, and then these other patients here. And Therese is very excited about this MRD sample here, where, you know, we don't have much expression of anything down in that sort of pre-B cell compartment, but we're seeing, you know, expression of the fusion in some other cell types and in B cells. So we're quite interested to dig into these cells and see, you know, what they're telling us about the biology of this sample and what it could mean for sort of MRD tracking in the future. So just to finish up, now that we can sort of reliably tag fusions in single cell and sequence lots and lots of cells to boot, what biological questions can we start to answer? Well, firstly, because we can now identify the leukemic subclones that express a specific fusion by sequencing multiple disease time points, we can start to understand how specific clones transcriptionally evolved during disease progression. That's what I'm showing here, just a heat map of the difference in expression between diagnosis and relapse of this particular fusion. And for cells that we know express the fusion, and then sort of doing a differential expression analysis and sort of seeing what other genes are sort of different there. We also had an intriguing observation in some patients that oncogenic fusions can be detected in non-B cell populations, and this may provide insight into the cell of origin and suggest that the initiating event occurred much earlier in hematopoietic development than we previously thought. So this is our control CRLF patient, where you see most of the fusions expressed just in the pre-B cells. And in this unusual patient, we do see the fusion expressed in other cell type lineages, which fit with sort of other clinical details of the patient. So it's very exciting to be able to see this at the single cell level. And finally, detecting residual leukemic cells that persist post-chemotherapy, which is also known as MRD, using single cell has been previously really, really difficult, as this constitutes such a minor population. And this approach has allowed us to successfully identify fusion expressing cells in this MRD bone marrow sample here. And it's significant because these MRD cells are the ones that eventually lead to relapse, and we can now better understand their biology using this approach. So we're very, very excited to continue the analysis of this data and see what it tells us. And I'll just like to finish up by acknowledging everyone who's been involved in this project. In our lab at Peter Mac, Victoria Streeton-Cook, who's the RA who was responsible for writing the software for the design of the fusion probes. And of course, Teresa Sadras from ONJ, who's been our wet lab collaborator, you know, help us source all the samples and interpret the biology. So it's been great working with her. And finally, just the molecular genomics core at Peter Mac, who helped us who ran all these samples for us on the 10X. So I'll just finish up there, and happy to take any questions.
Fantastic, Jovana. That was honestly an amazing presentation. I really, really enjoyed that. This is the second time I've got to hear this time with a bit more updated data. So it was great to see what happened when you put higher probe concentrations in. Great to see some of the biological meaningfulness of actually going through this work in the first place and getting it to a robust level that you can start having these patient insights. I mean, this is really, really powerful stuff, and hopefully it resonates with the audience here. I put a thing in the chat at the start. Sorry, I forgot to mention at the beginning, but David also just put a message in there as well. If you've got any questions, feel free to add them in the chat. I think David may even have the ability to take people off mute as well if you want to ask in person. So there is a question from Wei Shi, who asked, and I'm happy to help sort of jump in with this answer too. Sorry, it's hard to read this. Is it possible to detect fusion circular RNAs, either using Flex or the spatial transcriptomics platforms from 10X as well?
Someone did actually ask me the same question at AGTA, and we had a good chat, and I was very encouraging. I think it's a similar sort of signal, like you're essentially trying to detect this kind of like breakpoint. I mean, I didn't know anything about circular RNAs, but it seemed to me like a similar sort of problem as to what we were doing. So if that signal is unique to that population of transcripts essentially, then I don't see why you wouldn't be able to do it unless there are some other limitations around the biology there. But I don't know if you have any comments, Jeff.
Yeah, I think I'd reflect that exact sentiment. And in fact, I'm not going to give this away, but there is a researcher locally who's looking into this as well. It really depends on the context of that back splicing junction on the circular RNA that realistically is probably your only place to land a probe. If the circular RNA has a fusion within the circular RNA itself, of course, you can target that fusion just like you can in any other fusion. But if you're just after the presence or absence of that circular RNA, it's really going to be through that back splice junction where you're going to be able to detect that. So it's all similar rules to what's laid out in our tech note, to what was presented here. Could you do it on a spatial platform? Most certainly as well. So to add a little bit extra context there, what we saw here was obviously single cell, where we have two 25 base pair probes that land on a target, and then we ligate that together to form one 50 base pair probe. In our spatial platform, Visium HD, it's essentially almost identical chemistry. And there's two 25 probes that bind next to each other, and we ligate them together. And then our Xenium in situ platform, very similar yet again. And this time it's two 20 base pair probes. Well, it's actually a one linear molecule that has binding regions on both ends, but nevertheless, that's a 20 base pair binding region for each end, which equals 40 base pairs. And again, you ligate that together. So in principle, I don't see why it's not possible. But like all things, it's untested by us, which is why we were always happy to come and support your research in whatever way we can to help get this up and running. All right, no other questions, but I do have a question, and we kind of chatted about this a little bit before the seminar kicked off. But I'm a researcher. I'm really interested in adding probes. 10X Genomics provides a technical note, which really gives you some guidance on what to add to the probes in order for it to be detectable in the assay. But ultimately, where the probes bind, that design the onus is up to you as a researcher. So how do you even go about tackling that question? Can you use tools like you do for simple PCR primer design, or do you have to go and develop your own software package?
So as I alluded in my talk, we did have to write our own software to do this, and we will make that available when we publish this work. So anyone will be able to grab our package and download it and design probes to their own favorite fusion. So yes, we tried to basically synthesize all of the recommendations and the knowledge that we got from Calandra's work that I mentioned to try and kind of come up with some heuristic model around how to best design probes that were most likely to work for us. I'm a computational researcher. I don't work in the lab, and so we don't often. I've never generated data in my life before, so this was it's kind of scary for me, I must admit. But so we wanted to give ourselves the best chance of these probes succeeding. And so I guess we spent a lot of time thinking about, you know, what were the pitfalls, and sort of then building that into our software. And it's specific to fusions, but I don't see why you couldn't do that for any kind of maybe structural, expressed structural rearrangement that maybe you're interested in, like some sort of insertions or deletions or anything like that, that gives you that kind of very specific target that a probe could be very good at discriminating from the wild type, say. And it'd just be about sort of knowing what, you know, understanding the biology of that region and knowing the sequence, and then sort of being able to how to optimize for that along with all the other factors. But yeah, I think, you know, we're very excited about the possibilities of this and already thinking about maybe other things we could do. You know, could you look at some strange splice variants that come up in things that are not in the standard panel, but if you could sort of discriminate on some sort of splice junction that's a bit odd if you've got weird transcripts and things like that. So yeah, I think but yeah, it's custom software, so there isn't anything available yet. But lots of guidance there.
Fantastic. And yeah, for anybody who's thinking about this, 10x could be involved in any sort of conversations at the beginning, planning stages, to help arm you with the best tools that we have at our disposal for you to go and do this yourself. And beyond fusions as well, you mentioned other weird species. I should note that I have had people do this for, say, viral transcripts is another common reason to spike in viral probes in both our single cell and spatial products. Bacterial transcripts, to some degree, I have seen it done successfully in both single cell as well as spatial platforms too. I know we're at time. I have one final question for you, and that was around probe specificity. So did you observe any, or how, I guess, did you observe any off-target binding of these probes, and were you able to even sort of quantitate that? Did you have a feeling, a sense for that at all?
So we did. I didn't show the data here, but like I said, for the first experiment, we basically put the same probe in every sample, and we knew which fusions should be coming up where, basically. And like I said, we weren't seeing any MCF7 stuff coming up. Sorry, we weren't seeing any patient fusions coming up in our MCF7, but we did see some MCF7 fusion expression across our patient samples, which we found really interesting. And we thought that this must be some sort of ambient signal. And we have our PhD student now looking at this, and her first sort of preliminary work is sort of suggesting that yes, it is, because we can see this in the empty drops. So it must be something that's happening after pooling. We think that there's some residual things floating around, because they're actually being assigned to the wrong sample, if that makes sense.
Yeah.
Like, so they're getting a barcode from a sample that doesn't belong to them.
Yep.
To the sample. So yeah, when you put the probes into the sample with the sample barcode, they go into the 4 individual jars. And so, anything that should be picking up MCF7 should just have the barcode for the MCF7. But we're now seeing this assigned to the patient. And we're thinking this must be happening somewhere downstream in some sort of residual binding going on. And it's quite low level. And it's a very interesting signal that I think, but yeah, it's definitely ambient sort of RNA. But it was quite a cool sort of experiment in that we could actually pick this up and sort of say, okay, well, we know this signal shouldn't be there, so what is it? And yeah.
Yeah, I really like that. I quite often get asked about custom probes. Well, how do I know this is going to work? And our answer to that is, we don't really have upfront QC tools for you to assess the probes other than running the assay. But equally on the back end, I think it's also really important to keep some controls in mind to make sure this signal that you are seeing is real. I think we see it time and time again across multiple sort of single cell experiments. You want to make sure that these biological inferences that you're making are based on the true biology and not an artifact that's arisen, particularly when you start putting in probes that you've designed yourself. So I really like that sort of controlled manner that you've got here to be able to assess, okay, is this low level background signal background, or is it real? Or have you discovered something interesting about the biology you just didn't know because you're looking at it in a new way? So I enjoy that. So With that, I didn't see any other questions. Really appreciate both Jovana presenting today, a fantastic presentation, and everybody else for joining us online. And I'll let everyone go back to their Fridays and have a great weekend. Thank you.
Thanks, everyone.